Welcome to Catalyst Medhub, where we ignite understanding and make learning easier. In our today's class, we shall be discussing on the primitive streak. As we all know, primitive streak is just a streak that appears on the caudal end of the embryonic disc at the beginning of the third week after fertilization. And the appearance of the primitive streak is very important because it initiates many embryological processes such as gastrulation, that is the formation of the three germ layer, also induce the notochord formation. So the knowledge of primitive streak is very important. Now, what is primitive streak? Primitive streak is just a linear thickening band of a cell that are located on the caudal end of the dorsal aspect of the embryonic disc, and it appears on the day 15 after fertilization. So the key things we need to take note of is that first, it is linear. It is a linear band or longitudinal band. It is located at the caudal end of the embryonic disc, and it is present on the dorsal surface of the embryonic disc. Also, it appears 15 days after fertilization. So these are the things we need to take note of. We should know that it is a linear band thickening of a cell that is located at the caudal end, dorsal surface of the embryonic disc, and it appears 15 days after fertilization. After the appearance of this primitive streak, it can be very easy to know the axis of the embryo, such as the dorsoventral, the right and the left half, and the craniocaudal axis. So appearance of the primitive streak is very important during embryologic processes, and the primitive streak is formed as a result of the proliferation and migration of the cell of the epiblast. At the beginning of the third week of development, the cell of the epiblast begins to proliferate and migrate. So as a result of this migration and their proliferation, it leads to the formation of the primitive streak at the caudal end. The features of the primitive streak also include: one, there is primitive node, also called Ensign's node. Two, there is primitive pit, and three, there is primitive groove. So the primitive node is the cranial end of the primitive streak. The cranial end of the primitive streak is the primitive node, and it also occurs as a result of the thickening of the epiblast cells. It is also called the Ensign's node. The primitive pit is located, it's just a small groove that is located on the primitive node. So the primitive pit is located on the primitive node, a small groove located on the primitive node. I would say that the primitive node is the cranial end of the primitive streak. Now, the primitive groove, it extends throughout the extent of the primitive streak. It's just a groove that extends throughout the extent of the primitive streak. And both the primitive pit and the primitive groove also arises as a result of the proliferation and thickening of the cells of the epiblast. So from here, this is the cranial end of the embryo, and this is the caudal end of the embryo. We are looking at the embryo now from the dorsal aspect of it, because we said the primitive streak is located on the dorsal aspect of it. So the caudal end here, this is the cloacal membrane, and up here is procaudal plate. So at the caudal end here, we'll be having the primitive streak. So this linear band of thickening of the cell here is our primitive streak. I will say that the cranial end of the primitive streak is the primitive node. So this area here is the primitive node, and on top of small group that is located on the primitive node is called the primitive pit. So inside the primitive node there is a small group called the primitive pit. And lastly, the group that extend in the primitive streak is called the primitive groove. So this diagram here is showing us the primitive streak, the primitive node, the primitive pit, and the primitive groove. Next thing is the function of the primitive streak. Functions of primitive streak. 1. Primitive streak forms intra-embryonic mesoderm. Mesoderm. 2. It initiates formation of the notochord. Formation of notochord. 3. It demarcates the embryo into right and left half. It demarcates embryo into right and left halves. Then another important function of the primitive streak again is that it initiates gastrulation. It initiates gastrulation. That is the formation of trilaminar germ disc. 5. It also gives rise to septum transversum. Septum transversum, which forms the central tendon of the diaphragm. Also, appearance of the primitive streak determines the cranial caudal axis of the embryo. 7. It also helps to determine the dorsoventral surface of the embryo. So that is the functions of the primitive streak. Next is the fate of the primitive streak. Fate of primitive streak. The fate of the primitive streak is that it actively gives rise to the intra-embryonic mesoderm by the ingression of its cells up to the end of the 3rd week. Now, after the end of the 3rd week, it now begins to regress craniocaudally, that is from the cranial end to the caudal end. It will regress craniocaudally and finally disappear or become insignificant at the end of the 4th week. So, the primitive streak disappears or becomes insignificant at the end of 4th week. So that is the fate of the primitive streak. It gives rise to the intra-embryonic mesoderm due to the regression of its cells. And then from there it will now regresses, and by the end of the 4th week it will become insignificant or it will disappear completely. So next is the clinical correlation. It usually arises if the primitive streak fails to disappear at the end of the 4th week. If it fails to disappear at the end of the 1st week at the caudal end, it will develop a tumor which is called sacrococcygeal teratoma. So it's just a tumor that is formed as a result of the failure of the primitive streak to disappear at the end of the 4th week. and usually appears at the coccygeal region. It's all on the primitive streak. Kindly subscribe, like, comment and share. Don't forget to turn on your notification bell so that you don't miss any